Welcome to Academic Talks with this week's guest, Elçin Ünal. Academic Talks is a concept that we aims to have conversations with scientists we admire. Dear Ünal is an inspirational scientist with great scientific accomplishments. Currently, she's an assistant professor at UC Berkeley, Molecular and Cell Biology Department. In her words, they are interested in understanding the principles and regulation of meiotic differentiation. She is one of the most inspiring and enthusiastic biologists that come out of this country, Turkey. Ymen. We are greatly honored and thrilled to have you with us. Thank you for coming and joining us. So, you want to say something before we move on to the questions?
Thanks for the introduction. Yeah, I'm happy to be here and answer any of your guys' questions. Yeah, I've basically moved to Turkey like right after to US. Basically right after I finished college. So kind of like perspectives that you may be interested in if you're interested in sort of studying abroad or doing any postgraduate work. I think I may have some, it could be outdated information, but I'll have some information for you. But yeah, glad to be here and yeah, happy to talk to you guys.
Thank you very much. I have questions about it, so I will ask that questions later. So I'm starting with my first question. It's a general question. How did you choose your search topic? Like, if you had the second chance, would you change your area topic or specialize in different areas, or choose this one?
I think it's kind of in certain ways serendipitous, but like in certain ways it's sort of like life experience. I think if I were to take the similar research path, probably end up in here still. But it depends. So basically, when I started graduate school, I was very excited to do research, but I knew very little in terms of how to do research, right? So then come to grad school, you do rotations in different labs to pick a PhD lab. And what I liked about my project and the organism that I was working at the time and still currently budding yeast is it's very fast. You can actually design a lot of very elaborate genetic experiments, and that I liked a lot because there's a lot of like logic built into it, and so you can test it. It's almost like a playground, right? So you can come up with an idea and like, how am I going to test this? And then you can design the experiment in a very controlled manner. So that part I really liked. In terms of the actual like research phenomenon, it was actually quite serendipitous the way I started working on chromosomes and DNA damage repair. Long story short, I ended up there and it was fun because it was very, the field at the time was super, in certain ways competitive, but at the same time, everything is like happening super fast. So it was really, really engaging. And then being interested in chromosomes and working on DNA damage, then I got interested in meiosis because there you have extreme chromosome dynamics and the cells are literally like shedding their chromosomes, right? So that was very, very interesting. But then also I was in this embryology department as a yeast lab. It's kind of a funny sort of connection, but I would hear a lot about like germ cell biology and, you know, like how germ cells are special, and obviously meiosis happens in germ cells, right? So, but I didn't necessarily want to like switch organisms per se. I wanted to see whether there's a question that I can answer using a very simple model still. And that kind of took me in terms of being interested in germ cell quality control and aging, trying to ask this sort of kind of bizarre questions that are out of the box, still using the simple model system. But it's probably like, if I were to start now, it would probably be different, right? Because, you know, it's kind of hard to sort of, I don't imagine that, like, I think what makes research fun is you find some kind of an engaging question, but it could be whatever type of question. I'm not sure if like being married to the exact same topic is that healthy anyway. So I think it will probably be something different. And depending on like the circumstances, the mentor, the lab environment, and what's going on these days will probably determine. Yeah.
I think you are asking great questions. When I look at your articles, research, I think you're amazing at it. So one of your interests is about aging. I have a question about it. What can we learn from gametogenesis about aging? And do you think your work can answer and find a possible solution for how to reverse the effects of cell senescence and stop aging?
Yeah, I think I want to say we can probably learn a good amount since it has not been studied that much in this context. But I find that what is amazing is basically the meiosis or gametogenesis, right? The process that combines both the differentiation part of meiosis and meiosis as a cell division. It has kind of an inherent capability to remove age-induced damage, right? The cells basically evolved a way to fix themselves. So they're kind of creating new life, right? While they're getting rid of crap that the cell has built over time as it undergoes senescence. So what is the striking part of it is natural, right? You're not actually need to perturbate the system. One thing you can do is you can actually try to observe and get as much mechanistic information as you can by simply looking at it. But at the same time, I think what the neat part is you can actually do some forms of engineering as well, because if you understand the different components of how, different types of cellular damage are handled during meiosis, then you can try to build them outside the context of meiosis. And some of the work I did as a postdoc and currently now we're doing follow-up screens on it, suggested that certain meiotic transcription factors actually, when you express in mitotic cells that are undergone senescence, is able to extend their lifespan. So that basically tells us that parts of the meiotic program can be recapitulated outside of meiosis. And that kind of gets back to the other question that you asked, can we actually do something? Potentially, yes. But again, I think it's a sort of a analogy to, if you're a car mechanic, you probably want to learn a lot about the car before you start like fixing random parts of it and just kind of like anticipating, hearing a noise. I think that's what's wrong. But we need to know about it first of all. So we're kind of doing, in my lab now, parallel experiments where we're not only looking at the cells as they naturally undergo meiosis, doing a lot of live cell imaging combined with molecular genetics and genomics experiments to ask what happens, right? And what do actually cells do to their mitochondria, to their endoplasmic reticulum, right? To their vacuoles or lysosomes, to their, nuclei, like how does it behave, right? And do we actually see changes if we were to look at young versus age cells when they do go through meiosis? How do age cells handle that damage, right? So that's like one aspect of studying it. But in parallel, what we're then doing is, can we actually take meiotic genes and express them in an otherwise non-meiotic cells, like vegetatively dividing mitotic cells, and can we actually see any beneficial effects in terms of lifespan extension or changes in any of these like aging, biomarkers? So the answer is hopefully yes, but probably it's going to be taking a while, right? So we just started, So yeah.
Sounds really exciting. So, forgive me if I'm being like too basic, but I have a question. How do the cells solve the telomere issue, like telomere shortening issue during gametogenesis? Can protection mechanisms be implemented to pluripotent cells to prevent cancer?
I mean, telomere shortening is one way, right? So in the case for the stem cells, actually, they do have, continuous activity of telomerase enzyme, right? So that's why they don't sort of like go through this Hayflick limit. But that's only one of the ways. And I think why it gained popularity is probably because that was one of the first like observations. And while it's very interesting, it's not the only thing that happens in the cells. So if I were to sort of look through like our organism of choice, there are actually, in addition to, telomerase dependent, prevention of attrition of chromosome ends, yeast cells can also do homologous recombination mediated like lengthening. So they actually have backup mechanisms to sort of restore length. And in that sense, the telomere length itself does not seem to be a limiting factor, right? So I think for somatic cells, because they do have tight regulation of the telomere, right? Because they have a limited replicative potential. That's one thing that if one wants to be an immortal cell, needs to solve, but that's not the only thing that they need to fix. But so in terms of the meiosis part, though, we haven't looked that detailed in my lab, but I mean, other work has shown that, in addition to telomerase-mediated extension, there are recombination-mediated ways to sort of extend the telomere. So there are sort of backup ways to do if other things go wrong.
Perfect. So last week we had Orbide Gray as a guest. Maybe you know it. You know him. He's a.
I know of him. Yeah, he's a.
He works aging. Okay. So he says that he very proudly says that in the next 20-25 years there is a 50-50 chance that we will have the first anti-aging treatments. Do you think scientists should give an estimated time for these treatments or not?
I don't think scientists should give estimated time for anything, right? So if you remember, I mean, back in the day, Nixon said, war against cancer, we're going to cure it. All right, like that was a while ago, right? Have we cured it? I mean, biology is always more complex, right? And this kind of timelines of simplification is just, to me, a little bit, it can make the headlines, but whether or not they actually are realistic is another question, right? So while I think, I mean, people are like using, certain, so the other question is sort of aging field, for a while, right, they was sort of this understanding that the goal was to make everyone live very, very long. I don't know about you guys. I don't want to live that long. But what would be nice is whatever you live, you actually have a high quality of life, right? So then from that on, now it's more about health span rather than lifespan. Health span meaning, can you actually live in the amount of time you're living a life that, you have a reduced, risk of disease, you have a high quality of, life, right? That is much more attractive. And also I think something that could be more feasible than like being immortal. And I think, we can't probably like overcome the sort of rules of evolution, right? So that's like, again, I think it will be speculative to sort of make these sort of claims. There's like this much chance or in this many years, this is going to happen. I think we can probably talk about possibilities and sort of what are the viable like things that can be, thought about or gone after and what makes sense and what is sort of ethically actually reasonable to. But I'm not a big fan of like giving exact dates because science, if you have started doing it, there's a lot of gray areas and there's a lot of unpredictable nature of it, right? And you can't just like, it's not like, you have a scheduled test and like it's going to end on that date. It doesn't work that way. Yeah. I don't know if
your answer is perfectly reasonable. So another question. Due to aging, women's menstrual periods stop permanently. So when they are in a post-menopausal stage, with the decrease of estrogen and other hormones, they might have some health problems, heart diseases, diabetes, hypertension, and even fatty liver syndrome. Can be seen with these changes. Could these effects and the body damage be reversible if the patient gets in vitro converted or artificial gametocytes and be reproductive again?
That's an interesting question. I think there's some interesting work, actually. It's a little bit of a different thing I'm going to tell, but maybe it will tie back, is some of the earlier work in the worm, C. elegans, right? That's one of the sort of key organisms that has provided a lot of great insights into the biology of aging. And what they found in that case is basically, if you were to actually remove the germ cells, right? So if basically remove from the worm so that they can't make, oocytes, they can't make sperm. And what they found is when you do that early on, the organisms actually live much longer. So this sort of suggests some form of a trade-off between where you put your resources, right? Are you sort of, if you're going to go and make the germline, that's basically what sets for the next generation, comes at the expense of actually keeping your somatic cells or the soma fit. And so this like antagonistic pleiotropy is sort of this sort of term in general to sort of describe the relationship between these two sort of different lineages, right? When you're asking in terms of would there be, if we were to maintain, prevent basically menopause, long term is probably, I mean, if as long as you can maintain like, you know, levels of the estrogen, progesterone, and all other hormones in line, probably you will have more benefits. Whether or not that will also like end up causing more complications about like the risk of like getting, you know, ovarian cancer, breast cancer, because that's, you know, also I think a pretty decent possibility, right? I'm not sure in terms of like the intervention, what kind of a thing would be. Again, this is more of a question about, yeah, what kind of a, would you want to like do any kind of a systematic change versus you want to have a little bit more targeted, like, you know, targeted therapy, right? Because I'm not sure, you know, trying to like change something that is actually like, because the oocytes, you know, they are basically a set number of cells that are produced while a woman is an embryo, right? While it's in the womb, like embryonic, whatever, 9.5, they do produce like a set number of oocytes, and that's the pool they have, right? So while like kind of thinking through how you can like expand that pool, or whether or not you can play with like oocyte attrition rate and things like that, could theoretically be possible. I'm not sure if I would like to go there would be my answer, but that's an interesting thought.
That's great. So a month ago, we had another guest, maybe you know him, Randy Sheckman.
Oh, he's my colleague, yeah.
And actually, he suggested you as a guest. Maybe you don't know it, but yeah, we are so
we're in
happy.
the same department. I'm probably one of the few Turkish that he knows. He's great. I like Randy.
Great, and he has some strong opinions about publishers. What's your standpoint on that? Like, I wonder.
Yeah, I mean, publishing, I think, needs, and it's evolving, right? I mean, the kind of challenges there are right now, I think, if you just take biological fields as an example, right, and think about like the kind of publications you read 60 years ago versus now, the breadth, like how many different types of subfields and different, you know, ways of like doing biological research has evolved, right? So in the day, I think back, it was more of I don't want to say a simple task, but it was more, because less was known in terms of the expertise that you would need to review a paper, was not as extensive as currently, right? Right now, like if I get a paper from neurobiology, there's no way I have the enough expertise to understand, right, and evaluate critically what's going on. And because of that, I think there was an expansion of a bunch of journals, right? And similarly, there were expansion of, you know, the requirement for editors and all of that. But then I think what the kind of thing that has happened, which I'm not sure if it's like the right metric, is this metric of impact factor, right? That, you know, people use in general, because it's a simple way of using it, like, you know, whether or not your publication is worth by using the impact factor number for a journal as a basis. Yeah, and I'm not sure whether I agree with that. And the other thing I guess don't agree is, I don't agree that like publications should be, you know, you should pay money to read a paper, right? That's, you know, if you're using taxpayers' money, which is majority of the research in US, you know, is paid by the, you know, National Institute of Health, Sağlık Bakanlığı, right? That's like, you cannot, and then you basically pay to get published, right? You pay a journal a fee, and then they charge a fee to the others for them to read. And that's just sort of like a for-profit cycle for this kind of a work. I don't think it's appropriate. That's why I think I am, you know, definitely much in favor of publishing in open access, publishing in society journals where the editorialship is strong and transparent. Because transparency in review, I think, is also very important. Because you can say a lot of maybe nasty things behind the curtains, but when you know the two other reviewers that are also reviewing that paper, you probably would like to write something a little bit more constructive, right? Because at the end of the day, I mean, what is the job of peer review, right? If you think about it, the job is basically to cover blind spots, right? To see actually whether there's any part that has been missed in making those major conclusions in a paper, rather than finding a way to really trash that paper, right? So with that in mind, I think transparent review really adds a lot. Open access reads a lot, because even there's one person out there who would benefit from that publication if they were to have free access to it. That's totally worth it, right? You hear these stories like people get a particular, genetic disorder or find diagnosed with something, and they start digging the literature themselves to find out ways to help themselves or a loved one. And if you're restricting this kind of access to publication, that's not the way to go after, right? And promote sort of scientific prosperity, I would say. That's my thought. I think Randy and I probably align, and I've heard him a lot talking about these things, and he was the editor of eLife, as you guys probably know, for a long time before he stepped down. But yeah, I do share, I do share most of his views, I would say, and agree, yeah.
I totally agree with you. We have some questions in YouTube chat, so I can ask.
Sure, I
must, no problem. Do you think that CRISPR free edition of mitochondria and manipulation of orthogonal central dogma gonna open a new door in the aging studies?
So can you repeat? Is the CRISPR editing of...
do you think that CRISPR free edition of mitochondria and manipulation of orthogonal central dogma gonna open a new door in the aging studies?
Yeah, I think so. I mean, this type of, it will open new doors in any field in biology, right? Not only aging, but to be able to manipulate genomes at will, albeit be nuclear, mitochondrial, whatever. I mean, that's like a huge deal, right? And the fact that you can do in any organism that you can try so far and test, it opens up ways to study new biological phenomena, because you can look at much more rich biological space, but also it enables you to, do a lot of changes that kind of I dreamed of. The year I entered college, that was 1997. The Dolly, the sheep, I don't know if you guys probably know about it. It got cloned, and I was just like, oh my God, this is like the most craziest thing ever, right? And still it is, right? But you think about just from that time, that was 1997 to now. And even when you start reading, when actually the CRISPR field in terms of the biological part was building up, right? You have all these papers in terms of this very sort of detective story of what is this bacteria doing, right? What are all these interspecies, DNA sequences, all of that then turn into something actually applicable to technology, and then turns back and now you can treat disease like sickle cell anemia in Mongo. That's incredible, right? So it will definitely impact aging. It will impact a lot of things. It just needs to be used carefully too, right? I don't know if that was the question, or maybe I distracted. I heard Chris.
That's totally fine. One more question about open access. What about open access? Are you sensitive to publishing? Yeah, you answered this question, so we can move on.
Yeah, open access and preprint. I'm all pro for it too. Publishing, preprint.
When I check your publications, most of your papers are in open access, so I think it's intentional, right?
Yeah, it is intentional for sure. Absolutely. Since I run my own lab, I can do things the way I want. So it's very intentional.
That's really perfect. We are an iGEM team, and we have a project this year. So we have a 3 questions about our project. So I'm starting with them. You are doing dynamic imaging to build gene regulation networks, right? What can be improved in terms of microscopic work to imaging these networks? For example, did you face any technical problems with fluorescence microscopy while imaging gametogenesis?
Yeah, definitely. I mean, I think the one main issue, I mean, there are, I think, a couple of issues. One is, I mean, with the scopes that we use, we work at the limit of resolution, right? None of them has like super resolution microscopy, but in terms of being able to get information for things we're interested at a first glance, it actually is good enough. And it provides a lot of valuable information. I think the main challenge was dealing with phototoxicity and over time, because especially the starting point to enter meiosis is a time where the cells are especially very sensitive to light. So we really had to do, by we meaning my students in the lab, they did a lot of incredible like troubleshooting to optimize the condition that enabled us to do imaging over the entire course of meiosis. So we had to, you know, play with light sources, filters, and things like that. So it's a bit more like technical things. But at the end, the challenge was, yeah, to overcome phototoxicity and being able, because the movies that, you know, if you guys have seen any, those are basically taken over like 20 hours of time, sometimes more. And the part that is the most sort of vulnerable or sensitive is the part that of the entry, like when cells are really like kind of making the decision whether or not they want to go into the meiotic fate or not. So that was, I think, the yeah. Sorry, I have my little cat here jumping. Okay. Yeah, so that was the part. And in terms of sort of, yeah, but what we look at most with live imaging is organelle like inheritance and organelle dynamics. I think when it comes to gene expression regulation, we do some dynamic stuff, but most of that is now molecular biology and sort of like more genomics-y work. But yeah.
I will talk about our project in a second. Before that, I have one more question. Also,
Sure.
on the educational side, do you create movies from the images you take, or are you planning to integrate that part into your research soon?
Say it again. So do I plan to...
Do you create movies from the images you take, or are you planning to integrate that part into your research soon?
We do create... Sorry, the movies, like movies that, sorry, the time-lapse movies you're talking about on the microscope.
Yeah.
Yeah, so we do have that. Like basically anytime you do a time-lapse imaging, you have like in the order of like terabyte-ish, you know, like it's just, we fill, you know, lots of hard drives and storage, very, very continuous. So every time you actually get a movie, because what you're doing is you're taking snapshots of the same location, right, at a certain like time interval. So then you can either display them as montage, so like series of like images, or you can actually composite them. So one thing that's kind of nice is if we were to, you know, look at the online versions for the publications, not the PDF format, the movies are all there. It's just when you convert it into a PDF, you just see like the little play button and you don't see the rest of it. But they're all there. In terms of, because probably our website is a little bit outdated, I have to admit. We may not have much
I don't think so.
Yeah.
I genuinely think that your website is great.
Oh, thank you.
Most don't have that.
Yeah, it may probably require some like updates, but it's, yeah, it is. Thank you. So, but yeah, we do basically have a lot of movies, and then whenever, you know, for any of the publications that do involve movies, not only there's like montages for the main figures, but there's like supplemental movies that is, you know, added to that shows the whole thing.
I believe you have some animations too.
Oh yeah, the Claymation you're asking. Yeah, that was a fun project. Okay, that was when I was in my postdoc. And then it was actually a collaborative work between me and a graduate student in Angelica Amon's lab where I did my postdoc. So we had this basically findings that were published in an eLife paper that was about how the misregulation of this particular cyclin CDK impacts meiotic chromosome segregation. So then at the time, actually, eLife kind of just launched. Maybe we were in their first or second issue. Actually, they don't have issues, but we were one of the like first papers that came out. But what the kind of thing is they ask, you know, do you want to provide anything that's more descriptive, promotional for your work? And then at the time, like the student was about to graduate, you know, earn his PhD. And then I was on the job market, meaning like interviewing in different places. So we had some time to burn. So we're like, oh, we should just do like this fun movie. So we like literally went to an art store, got a bunch of like clays and like watched movies in terms of how like people do this. Because you basically need to just have like a stable, like, you know, camera and just take pictures. And then we had a few other people help. That it was a lot of fun. It took like a whole afternoon to like make the actual like thing, and the assembly itself took another like half a day, but that was the end product. But it makes a good movie for like lectures and stuff. I have a lot of colleagues that use it for their like meiosis lectures, because yeah, the homologous, the crossovers and everything is kind of like shown accurately, at least for the wild type one. The mutant is definitely Baroque, but the other one is, yeah. So yeah. I'm not sure if I'll do. Maybe when I have some more time, I may get back into that. But none of my students, they're probably much more serious and busy with their work and not like time doing things. But I would like to.
It was very fun to watch. Thank you for that. So, about our project, we are interested in imaging developmental processes, and we are working on a project to make cells emit focused and more intense light. Think it like a better version of fluorescence microscopy. We are planning to create cell lasers. In this way, we hope to create better imaging modalities in developmental research. Could you describe some open questions in your research domain? We really appreciate some insights.
Okay. Sorry, tell me the last part, because this cat is scratching.
Okay.
I heard anything else you want to improve, like observe developmental processes and improve imaging. Okay, and then tell me the last part of your question.
Okay. We are planning to create cell lasers.
Okay.
In this way, we hope to create better imaging modalities in developmental research. Could you describe some open questions in your research domain? We really appreciate some insights.
Yeah, I mean, there's a lot of open questions. I think the one kind of open question that would be great is to be able to actually, for the case for, you know, meiosis and gametogenesis, you know, it would be so great if you actually can grow oocyte and sperm in dishes and like observe what's going on. I mean, there are actually really cool imaging that has been done primarily in oocytes in Melina Schu's lab. If you guys are looking for anyone else to invite, she's in one of the Max Planck. She's actually one of the only, I think, women directors of a Max Planck Institute. So she does a lot of mammalian oogenesis, and because its oocytes are huge, right, compared to other cell types, she can get incredible images. But at the same time, in terms of harvesting and the sort of timeline of doing this when you're harvesting stuff from actual organisms versus if you are able to grow things from stem cells in culture, that will be amazing. But I don't know when that will happen. That's why I still have a job, because for yeast, we don't care. Like, we can induce them to undergo meiosis anytime we want. That's one of the like best parts of it, because we can control it, right, in an environmentally sort of like regulated fashion. But I think that will be really, really cool. And then to be able to, observe things in addition to chromosomes and sort of really looking in terms of, what other aspects of, changes that occur in meiosis are actually conserved across evolution. That will be really, really cool. In terms of like developmental questions, right, trying to look at things in the context of a tissue in a 3D architecture and having like enough image resolution to do that, that's like really a key. And that's basically, one of the sort of very sort of, exciting areas of research. That's more like physics questions. But there are, I mean, I have colleagues that are solely focusing on microscopy, like Eric Betzig, who was the Nobel laureate for super resolution microscopy. Now he's doing all kinds of other. So we actually recruited him, not me, but Berkeley recruited him to a few years ago. And he's working on more sort of live imaging based technologies for super resolution. And so that's basically is the sort of next route of things, I think, to be able to look at things in 3D over time and as physiological conditions as possible.
Then thank you. Do you know anything about live cell lasing? Maybe can I skip this? Like
Live cell
lasing.
laser.
The thing
No,
I'm
sorry.
talking about lasing. Like we are getting lasing from live cells.
Uh-huh.
Laser cells. Do you know anything about them?
I'm not sure I understand what you're talking about. Maybe tell me a little bit. It may be a little jargon that I'm missing.
Okay. There is a question, but I wanted to import it. Okay, I'm just asking the question. Can you give us some information about live cell lasing and what kind of problems do you have when you try to observe living cells in your experiments? You answered second part of the question, so I didn't want to ask all the way through. So, yeah. Do you have any information about live cell lasing?
I don't know what you mean by like lasing. Sorry, am I like?
Yeah, no problem at all. No problem at all. It's totally. Thank you anyway. So we have some questions about your research topics. Your NDT-AD research is quite intuitive and important to me. And do you have any future plans about studying those genes in mice or even in human? Are you guys planning?
Yeah, it's probably not human anytime soon. But what we're doing is basically we're doing large-scale genetic screens for genes that are downstream of NDT-AD, initially in yeast, and then the next organism we'll be looking at is worms for the homologs. And then, yeah, so I think maybe hopefully someone else can do the mouse work. I'm not sure if I'll ever do mouse work. If it can be done in tissue culture, that will be fine. But I think C. elegans or flies would be the most complex I would get at, just for personal reasons.
But you said you are not interesting to increase your lifespan.
No.
But now, for now. But why? Why don't you want to study on mice, or why don't you want to increase your lifespan?
Why don't I want to study in mice?
Maybe.
Well, yeah, I think, yeah, I mean, that's for personal reasons, meaning I'm not very good at, yeah. So in terms of like working with animals, requires a lot of stacking of animals. So I think for me, it needs to be validated in a certain way. But I think I will be totally comfortable doing experiments in organisms that have less complex nervous systems and pain. But again, that's like a personal choice, right? In terms of like finding, studying something and being able to like test in either in like an organoid type model or a tissue culture model, that will be totally fine. And that I think could be relevant to do some sort of at the cellular level, something that you can test in a mammalian system. But I'm not sure if my lab will be doing organismal work in mouse per se. The other question, why don't I want to live longer? Because like,
maybe that's a
want to live.
Question. Yeah. Okay, you don't have to answer it.
Because I think it's to recycle and like bring back your carbon and nitrogen and everything back into the environment, so something better can come after you. I don't know. I mean, I don't think I think. Yeah, having a finite time is a good thing, right? That's like a very good certainty. That probably will, like, everybody's aware of that's going to happen to them one day.
Yeah, of course. There is a question in YouTube chat. What makes Berkeley one of the best institutes in the world on biology? What are the differences you observed between the institutes on Turkey and Berkeley?
Oh, okay. Yeah, so Berkeley is great in many ways. I think first, the thing that I like as a university, it's a huge campus. It has a lot of diverse students coming from all kinds of economical ethnic, racial, whatever backgrounds, right? So there's a lot of, so when you are in the campus, the first, like the feeling that at least I've gotten when I visited, I felt really, really small. And that was like really good. And you feel really insignificant because there's a lot going on around you. And there's like people are walking around, it's beautiful weather. And they are like into what they're doing, but not in a sort of condescending or an arrogant way, but really in a way that they care. So in general, there's a very great level of collegiality, a level of excitement, and a level of not taking yourself very seriously. Because I think that's something important to sort of bear in mind, that you can like talk to someone like a Nobel laureate Randy and joke, and make sort of funny jokes about him, and he doesn't care, and he's very down to earth. You can also be at a state that you can be, an undergrad, a graduate student, whoever on campus, and like reach out to someone. Well, it may take some time to hear back because usually faculty are busy responding to email. At the same time, I don't think there's this level of like hierarchy. That's, I think, really nice and liberating. So as a trainee, the hope is that you're not intimidated, so you're not afraid to state your opinion. And I think it's really important because, again, you guys are the next generation, so you cannot be silenced, right? I mean, on the contrary, we should listen to you more and more. So I think that type of atmosphere is sort of how it is here, and I really like that. And also I think it's a public university, even though it's expensive still relative to some other countries and standards. The fact that the school provides financial aid to students who cannot afford and really don't admit, we don't have like legacy admissions. I don't know if you guys know what that means. If you're rich, you can make it in. That's not a case in Berkeley. You actually need to like earn to get admitted to Berkeley. Whereas there could be other private institutions where, you can get admitted because you have, you have a healthy, you have like financially, you have, very wealthy parents, or, you have a certain endowment in the school, and all of that doesn't matter here. But the downside is we don't have that much endowment, right? In terms of money, we're obviously far behind Stanford or Harvard. But I think in terms of the quality of students, it's an amazing place, and the quality of colleagues. So in comparison to Turkey, I think what I would say the main difference probably is that the level of hierarchy is not there. There are, for example, like we have a department chair. Actually, there's 2 co-chairs, but it's not about like that seat is not indicative of power, but rather it's indicative of service. The person becomes a chair is basically they're doing favor to the rest of us by like, taking care of administrative duties rather than sort of being authoritarian. So that I think is really great, and I really like that aspect to it. I don't know if that makes.
I totally understand you. Yeah, I know the hierarchy, so I feel you. One more about your research interest. Your other research interest is Lutein mRNA. Am I right?
Yeah.
Can you talk about its importance? Can we use it as a tool like RNA interference? We used it like.
Yeah. No, that's a very interesting question. So I think what is its use, right? So the Lutein basically stands for long undecoded transcript isoform. And so what it is basically it's an mRNA molecule that the cell makes. But, based on all what we learned in textbook, an mRNA is supposed to make protein, right? Because it's a central dogma, right? DNA makes an intermediate messenger that it turns to protein. But in this case, what we found is actually you can actually the cell can make an mRNA, but the purpose of making that mRNA actually results in the opposite effect of turning down protein synthesis, right? Inhibiting protein synthesis. And you may ask, why would you want to do that, right? Or, I don't know the answer why, but I can speculate, right? And what I would say to that is, if you think about sort of gene regulation, the primary sort of effectors there are transcription factors, right? These are basically DNA binding proteins that have the ability to recruit RNA polymerase to generate messenger RNA. But what the Lutein mechanisms does is, depending on where a transcription factor binds, it can do either an activation or an inhibition when it comes to protein production, okay? So in a way, you use the same toolkit that you would normally use, transcription factor and ribosome, which I'll get maybe if I have time, to actually do both jobs. They can make protein, or they can actually inhibit protein synthesis. So in the case for the Lutein, what happens is that when a Lutein mRNA is being transcribed, so the cell makes this Lutein mRNA, but the mRNA itself cannot make protein. And the reason for that is the 5' leader region of the mRNA actually contains a number of upstream open reading frames. So these are UORs. So these are actually small regions, within the mRNA that can be translated, but they don't actually make productive peptides, right? What they do is basically think about them as competitive engagers with the translation initiation machinery and the ribosome. So the ribosome tries to decode this message, but all it does is basically generate these UORs, but it actually can never, it cannot efficiently translate the open reading frame that is embedded in the Lutein, okay? So at the mRNA level, it's a dead end product. But then what it does is, as it's being transcribed, the transcription event of the Lutein, because of the location of where it is relative to the promoter region that controls the protein coding mRNA isoform, it actually silences that downstream promoter. So as a result, you're making this mRNA that doesn't make protein, but as you make it, you actually silence the promoter of the mRNA that can make protein. So it sounds a little confusing, but in theory, when you actually think about it, it's a super elegant way to really use the same machinery, the ribosome and transcription factors, to either activate or inhibit gene expression. Because sort of like where they would bind in the genome would in a way dictate whether or not the cell is making protein or not. So it's a good way, I think, from the sort of evolutionary perspective or developmental sort of gene regulation perspective, to think about if you want to coordinate as you're making transitions from one cell state to another. If you want to, turn on a bunch of genes because they're required for that given state, there will also be genes whose expression is not beneficial for that particular cell state. So this mechanism, in a way, allows for doing this sort of coordinated repression and activation in a timely manner. I think that's kind of the answer. I don't know if that makes sense.
It's a bit confusing to me, but I will definitely follow up your work because I think it's very interesting. Another question: how did CRISPR and Ymen factors affect your gametogenesis related research? Did any of them help you achieve an unanswered question?
Yeah, I mean, the CRISPR, I mean, we use it in the lab for, making strains and, any kind of changes that you want to make to generate sort of whatever genetic toolkit you want to create, you can use CRISPR, right? So we use the sort of editing technology for that. In terms of Yamanaka factors, I think they have been shown, and that's not our work, but others, in that their expression actually can be associated with sort of like reversal of some of the like senescence factors. So there's work done by others in tissue culture context with the iPS cells that indicate that they have sort of age reversal effects, but it's not our work. Yeah.
Great. One last question, and then I can ask two more questions. It makes sense. Okay. This is a big one. Will scientists ever solve all gene regulation systems in nature and generate a synthetic organism with only desired regulatory mechanisms and genes? If we hypothetically accept the existence of such an organism, what sort of assistance would it give to scientists?
I see. I'm not sure if they will be able to. So the question is, I think, two parts, right? One is, will we be able to understand ever all the types of gene regulatory systems that occur in nature? The answer is, I think, if you look broad enough, you may, but I'm not sure whether or not, practically speaking, that's going to be feasible, right? Just think about the number of different organisms that are in the world, and there's obviously common features, but here and there, you definitely run into, unique modes of like regulation that, you may have missed in another context. So that, I think, can only be answered depending on how much depth and breadth people go in looking at across different biological systems. The other question is in terms of like whether or not you need to know all of that to actually build a synthetic organism. I'm not sure whether or not that's the case. And people have like made attempts, right? So, they did try building synthetic like genomes and synthetic organisms with, in the case for yeast and bacteria, but none of it is actually just starting from scratch, right? There's always some kind of a, you provide some of the basics of the cell, or, a cell with a membrane, and then you express, maybe like put all the chromosomes into one chromosome, or like modulate the telomeres and things like that. But we haven't like created life in the sense of how it started, that we don't know how it did, right, to begin with. So I don't know the answer to that question. Maybe would be the answer. But.
Great. So we have a question about Turkey. Turkey-related question, I say. Maybe, in Turkey, we don't have evolution in our curriculum. Maybe you've heard it.
Yes, I mean, I went to high school and undergrad in Turkey, right? I basically moved at the age of 22 to here. Yeah.
So, do you think it's possible or reasonable to understand biology without evolution, without understanding or hearing about evolution?
I mean, there is a famous saying, I'm going to butcher Dobzhansky, but basically, evolution makes what biology makes sense, right? So I think the answer to it is it will be difficult because, again, you don't necessarily need to maybe know to all extents of different evolutionary phenomenon and hypothesis and really like go very deep. But at the same time, if you don't integrate the notion of evolution and the fact of evolution into thinking about biology, I think it could be very, very difficult to sort of understand some of the underpinnings. And it's in a way a very sort of helpful and insightful thing to know, because it really like helps you put some understanding and logic into like how things work. And there's clearly like a lot of mysteries that we don't understand about biological systems, the living beings and unliving beings in general. But at the same time, thinking through the lens of evolution actually enables you to get maybe a little bit more coherent understanding of things. So I think it is important. And even if, you may not be getting it on lectures, I think it's good to read books. It's good to self-educate yourself. And the great thing is, we live in the age of technology now, right? So if you want, you can get that information, right? And it doesn't, you can actually, I wouldn't be bound by the rules that, of anywhere you are. In the same sense, like if you can go to college anywhere, you can be in a super, super liberal country, but self-education should never stop, right? So in that sense, it would have been great to have it as part of the curriculum. The fact that it's not there is unfortunate, but that should not prevent you guys from reading about it, learning about it, thinking about it, and judging it yourself, right?
I think that's a great answer. I have one more question. Can I ask it in Turkish? Is it
Sure.
okay?
Sure.
Okay, I'm asking. Son olarak bize önerebileceğiniz neler vardır? Toplantının başında dediğiniz gibi.
Son olarak önerebileceğim neler var? I mean, okay, Türkçe şimdi nasıl söyleyeceğim?
You can talk in English. It doesn't matter. It's totally fine.
So I think, let's see, I'm gonna let me speak a little bit English, and then I can like try to see if I can be coherent in Turkish. I think the important thing for you guys is, again, you're the next generation, right? That part. So you're the next generation. And I feel like the world right now is in not the best place, right? We can all agree. Everywhere in the world, chaos is there. You know, you look, you know, here, racism is here. I think racism is a lot of places, but manifests itself in different forms. And there's also the sort of issue about like what is actually factual information, right? Because the thing that is now is you can get your news from a lot of different sources, but that comes with the responsibility. Nobody tells you this is the right information. It really is determined by the sources that you pick to receive those news. What I will tell you is you're all scientists. Use your science judgment, the education that you get to really be skeptical and apply that to anywhere in life. And really kind of, for the facts or for the information that you're getting, use the same kind of scientific judgment and rationale in, you know, assessing the validity. Because once you have that skepticism built into you, I think, and if you have the goodwill to sort of do better in this world, the combination of that will be very, very helpful, right? And we need more people like that in this world. And instead of dividing people, we want people to be able to combine them, right? To actually unite, because, you know, that's what we need in the world right now. So I think do good science, keep on skepticism, and don't take things for granted. Is all I will say. I don't know if that makes any sense.
It makes perfect sense to me. So thank you for joining us.
Yeah, absolutely. I have a couple of minutes though. Let me just one moment, okay? I just want to do one thing because I 1 second. Okay. I think yeah. You can go ahead and ask. Yeah. Yeah. Yeah, absolutely. Yeah, so okay, the first question. Yeah, I actually, so I don't have a scientist in my family, no PhD in my family. So it was actually the high school teacher, biology teacher, that got me interested. So growing up, I thought I always loved animals and nature. So I was really like into sort of nature and biology, but I didn't think actually you can do biology as a profession. Okay, I was more thinking I will be a vet. That was kind of like me growing up. But then in high school, I really like all the STEM classes, like chemistry, math, and biology. Or physics I like, but not super much. But the biology teacher I had, he was great, and he didn't teach it in a way that was like memorization. It was more, you know, curiosity based, and you would like have more question at the end of the lecture. And he's the one at the time, Bilkent University, which is the school that I ended up going in for college, was just like starting. So he told me, you know, if you like genetics, if you like molecular biology, if you like this kind of stuff, like you should consider the school. And that's how literally like I like found out about it. And I'm like, and then I read more and things like that. So then when I got there, I mean, it was really nice. It was, you know, my class very small, right? Like every year they would admit at the time 15 students. So my cohort of classmates we were 13 people. And it was really nice in terms of the theoretical education, but research was difficult to conduct. The resources were not available that much. It will take time. And even though, we learn a lot, the sort of put that into practice was difficult. So what I did is basically, as I talked to, there were 2 other classes that, started before I entered. So I would basically ask the senior, students, like, what are you doing, afterwards, and things like that. And it turned out that all of them sort of followed these footsteps, that if they want to do research, they would apply to programs in Europe or the US. So in terms of master's versus PhD, so in the US, I think the traditional thing, so I did apply, like in my junior year, before the summer of my junior year, I, basically gathered a list of like 100 faculty, I think, all across US, Europe, read on their website, try to read for some of them their paper, 1 or 2, and email like every single one, really like custom emails, right? Not just like, dear professor, I'm interested in your work, do you have space? But just like write things that one person responded back, and he said, if you can pay your, expenses, you can have us, you can work in my lab. And that's literally how I came to US to do research in the summer. And in that case, I have to thank my dad, because I didn't have money at the time. I'm like, can you please pay for my ticket? And so that's how I ended up coming. And then I was fortunate enough to work with a postdoc who was extremely amazing, and she taught me a lot of things. And this particular professor, wrote me a good recommendation letter, and I basically ended up getting into graduate school in places that he actually has connections. And that's how like serendipitous it was. In terms of why PhD, because that's the main part that is in the US, right? So there's no a separate like master's and PhD program. Officially, like the first 2 years of your PhD, if you were to leave the program, you would get a master's degree after you pass your qualifying exam. But the other kind of obvious benefit is like you get paid instead of paying. And that's good, if you're not economically, if it's not available, then that's, a good way to kind of pay for your education. I mean, it's not a lot of money, but at the same time, you cover your living expenses. I mean, you can live, an okay time. And it is, again, temporary. And also, I mean, you're mostly a lot of busy, so there's not that much like opportunities to spend a lot of money. So it worked out in that manner. And I actually didn't, now that I look back, it probably would have worked in a different way, because it's just so random the way like the different things align, that I'm not sure if you can sort of repeat that same thing if you run the iteration one more time. But yeah, so it was that. I don't know. And in college, I would say, I was really good in high school, like valedictorian and all of that. But in college, the first 2 years, I like was terrible because I didn't study that much, and I was a little like, cocky, like, oh, I know it all. But like BCamp was really hard, right? And this people, my classmates, they were like really, really good. So not taking things seriously for the first 2 years, I definitely paid for it afterwards by just like studying a hell lot more. But yeah, it was that.
I believe we don't have any more questions. So thank you for joining us. I genuinely think that this was great. I learned much. I learned a lot. Sorry. Thank you for that. Thank you for joining us.
Yeah, you're welcome. And if you guys have any questions, I don't know, what year are you guys in, like, what year in college are you currently? Or are you already, yeah?
I'm sophomore.
Sophomore.
Yeah, first. Mine's first.
First, okay. Okay, so really early on. So if you have any questions down the line in terms of, doing internship in US, graduate school, blah, blah, blah, yeah, feel free to send me an email. I'll be happy to help if you want to pursue, academic research after graduating. I think a PhD is a good thing. It definitely it gives you earned confidence. So in that sense, minimally, even though you may not like, it's very unlikely that every PhD is gonna result in a groundbreaking discovery. Certainly mine didn't, but at the same time, it's an incredible thing to do in a way that going through the darkness on your own for a while and coming out of it in light and really like earning confidence. It really changes the way you evaluate things in life. So minimally for that, it's a great thing to do. And if you want to do it, let me know. I will try to provide some more information to you.
That's really amazing. Of course.
Sure, that would be great.
If you want to come back,
I was supposed to come back in the summer, and then I was like, just everything went to downward. So I'll definitely. Normally, I come every year, and and it's sometimes at a meeting either in Europe or in Turkey to the equation, but like this year, it definitely didn't happen. But yeah, I will be coming hopefully next year, sometime around the summer. But yeah.
Okay, then I think this is it. Thank you for taking your time.
You're welcome.
I really think this was great. So thank you for that.
Okay, so, glad
Thank
to
you.
hear that. Yeah, it was fun talking to you guys. Take care, okay?
Thank you.
See you.
See you then.
Ya
Thanks.
iyi geceler. Kusura bakmayın konuşuyorum fazla Türkçe ama hani Türkçe
Estağfurullah.
Okay.
Kendinize iyi bakın. Görüşürüz.
İyi akşamlar.
Sağ olun.
İyi akşamlar. Bye bye.
Bye bye.